Today we'll be talking about the HLA-DRB1 gene for our bioinformatics presentation. So this presentation is done by me, Helen, Xingmin, and Isaac. So what is the HLA-DRB1 gene? It resides at chromosome 6 and is part of the human leukocyte antigen class 2 family. The gene provides instruction for making protein that plays a critical role in the immune system. The gene is part of a family of genes called the human leukocyte antigen complex. This HLA complex, which is human leukocyte antigen, helps the immune system to distinguish the body's own protein from proteins made by foreign invaders, such as virus and bacteria. The genomic sequence length is 11,074 BP, consisting of 44.37% of GC and 55.62% of AT. So this shows the transcript sequence of the HLA-DRB1 gene. For the protein structure, we have used a cartoon to show its secondary structure and the superfamilies that are related to our gene of interest. So CATH shows the superfamily summary and sequence diversity.
So what's the purpose of our study? The variation in HLA-DRB1 gene have been associated with an increased risk of developing various autoimmune diseases such as Crohn's disease, rheumatoid arthritis, and type 1 diabetes. This could be due to the numerous mutation sites and the multiple natural variation of HLA-DRB1. These would translate into altered proteins, resulting in the immune system targeting what they deem to be foreign and ultimately giving rise to autoimmune diseases. So therapeutic drugs developed will target these altered proteins produced to treat or alleviate the symptoms of the diseases. It was established that animal models have been used to test the drug's effectiveness, and thus in this study, we are determining the animal species with HLA-DRB1 gene that has the closest resemblance to the human HLA-DRB1 gene, so it can be used as an animal model for the most accurate drug testing. This is especially important since there's a large population of people suffering from autoimmune diseases, thus it's vital to develop drugs highly effective against the disease. In the next slide, we can see the pathway of how when MH class mutates, it will lead to an alteration in cell adhesion which can disrupt important cellular processes and lead to a variety of diseases. The following slide, it can be seen how the KEGG pathway is shown to see how HLA-DRB1 gene mutate, which will lead to diabetes type 1, and how when HLA-DRB1 gene mutates, it will also lead to rheumatoid arthritis. So for bioinformatics analysis tools that we used, firstly we used NCBI to obtain all the sequences. After that, we did DNA and protein statistics to analyze the target genome and protein. Next, we used UniProt, which was to obtain information regarding the variants and mutation sites of the gene, and KEGG pathways allow us to view how the mutations will cause an immune response. Finally, both cluster omega and NCBI BLAST tools were used to compare the sequences of human HLA-DRB1 genes to the sequences of various species.
Okay, using UniProt, we are able to identify mutagenesis and the different variants of this gene. As you can see from the diagram, the gene undergoes some mutation, but there isn't a trend in the mutation sites. The gene also has many different natural variants, which may possibly be the reason for multiple diseases associated with the HLA-DRB1 gene. The diagram, next we can see the diagram, which shows different mutations of the gene and the specific base pair where the mutation occurs. Then the diagram above now shows the multiple variants of the gene. As you can see, there are many different variants to this gene, resulting in which may result in the formation of diseases associated with this gene. Okay, so using BLAST, we were able to find different species of animals that has the HLA-DRB1 gene similar to humans, and we have chosen dog, pig, chimpanzee, gorilla to further our research to find suitable animal model. Okay, So after finding the mRNA sequence of the desired species, we use cluster omega 2 to do global alignment of the different sequences. As you can see from the results, the DRB1 gene in chimpanzee and human is the most similar with 96.89% similarity. So to further test this, we have done local sequence alignment using BLAST to check the similarity between the genes.
So this print screen shows the local alignment using BLAST and the mRNA sequence of the gorilla. This is the result. And the next slide shows the local sequence alignment using BLAST for SARS-CoV, also known as the pig. The next slide shows the print screen for the local sequence alignment using BLAST for Canis lupus familiaris, also known as the dog. And the following slide is the print screen for local sequence alignment using BLAST again for pan troglodytes, also known as the chimpanzee. So in conclusion, when we compare the percentage identity match from doing local sequence alignment, BLAST, and when we compare the global alignments of all the sequences with the HLA-DRB1 gene using cluster omega, we reach the conclusion that it is best to use chimpanzees as animal models to test therapeutic drugs as they have the highest similarity to 97% local and 96.89% global with the human HLA-DRB1 gene. The next slide shows our references and sources. Thank you.